once again welcome to the class. Here we are going to discuss about the metabolism of pyrimidine nucleotide in the subject Biochemistry BP 203T. So, in the previous lecture we have discussed about what are nucleotides and the biosynthesis of purine nucleotide. In this lecture we are going to study about pyrimidine nucleotide. So, let us begin with the topic. So, there is a small revision of nucleotide. Nucleotides are compound which are composed of three things primarily, that is nitrogenous bases, purines and pyrimidine, pentose sugar and a phosphate group. So, phosphate group there can be 1, can be 2 and can be 3. If there is one phosphate group incorporated, that it will be called as monophosphate. If there are 2 phosphate groups are incorporated, that will be called as diphosphate, and if there are 3 phosphate groups incorporated, that it will be called as triphosphate. So, we have several compounds like adenosine monophosphate, adenosine diphosphate, adenosine triphosphate, AMP, ADP and ATP. We know ATP more frequently, we have studied about it. So, and they are the structural component of DNA and RNA. Now, we have already discussed about the sugars that are present in DNA and RNA. In DNA there is a deoxyribose sugar and in RNA there will be ribose sugar. So, what is the difference in deoxyribose and ribose sugar? You can see in the ribose sugar at the second position, at the second carbon, there is the presence of OH group, whereas in deoxyribose, as the name suggests, there is a less number of oxygen present in that sugar moiety and it is on the second position. There is a incorporation of only one hydrogen atom. So, today in this class we are going to study about pyrimidine nucleotide, its synthesis, the steps involved in the synthesis and the degradation of pyrimidine nucleotide. So, here is the structure, we do have the structure with us. We can see it is a pyrimidine nucleus, it has 2 nitrogens and there are 4 carbons in the structure. The third nitrogen came from glutamine, the second carbon, that is on the second position the carbon, it has came from our carbon dioxide, and rest of the molecules, that means from C4, that means we can say from N1 to C4, all the elements came from aspartate, another amino acid. So, here there are 2 amino acids, presence of 2 amino acids and 1 carbon dioxide. Let us see. Okay, one more thing, that is pyrimidines there are 3 pyrimidines, that is cytosine, thymine and uracil. Thymine specifically, the thing has to be remembered forever that thymine will be only in case of DNA, whereas RNA has uracil in it. Okay. So, we have cytosine in common, both DNA and RNA has cytosine in it, but thymine only is in DNA, whereas uracil will be in RNA. Clear? Now, let us see how the synthesis going on, how the synthesis starts. First of all, I would like to tell you that it is much more simpler process than our purine nucleotide synthesis, because there are only incorporation of 2 amino acid, that is aspartate and glutamine and 1 carbon dioxide. The whole thing contributes 1 pyrimidine nucleus. That is, pyrimidine nucleus is first synthesized and then attached to ribose 5-phosphate. So, here is not that we already have a ribose 5-phosphate and then we are incorporating our pyrimidine nucleus. Earlier in the purine nucleus, it was the thing that we were having ribose 5-phosphate from carbohydrate metabolism, and then we are incorporating, synthesizing and incorporating our purine nucleotide. Here the thing is different. We are preparing pyrimidine ring and then we attach that to ribose 5 phosphate. Okay. So, let us see the synthesis. This is the synthesis. First of all, let us start preparing our pyrimidine nucleus. So, for that we will be needing two things which we have seen earlier in the structure, that is carbon dioxide and glutamine. These two things with an enzyme, that is carbamoyl phosphate synthetase 2, there will be a synthesis of carbamoyl phosphate. So, by using two things, that is carbon dioxide and glutamine, we are using an enzyme carbamoyl phosphate synthetase 2, and there is a synthesis of carbamoyl phosphate, and there is an incorporation of your ATP molecule. ATP converted into ADP, that means ATP is consumed, 1 PPI, that is inorganic phosphate, 1 PI inorganic phosphate is consumed and we got our ADP. So, by converting the whole thing, that is CO2 plus glutamine, then we have our carbamoyl phosphate, which is then converted into carbamoyl aspartate. We know because there is a, in the name it is already there, aspartate. So, we have incorporated here what? Aspartate amino acid. Okay. So, we have here incorporation of aspartate, and the enzyme which we are using here is aspartate transcarbamylase. So, by using our aspartate transcarbamylase, we have converted our carbamoyl phosphate into carbamoyl aspartate. Then we converted our carbamoyl phosphate aspartate into dihydroorotate. This dihydroorotate, remember this name, it is very important. Rather, I can say this orotate after the conversion from dihydroorotate into orotate, that orotate is very important in this pyrimidine nucleotide biosynthesis and in the degradation product. Okay. Let us continue. That the conversion of carbamoyl aspartate into dihydroorotate is done with the enzyme dihydroorotase, and a water molecule is going out of the reaction, water molecule evolved. So, after the synthesis of dihydroorotate, it will convert it into orotate. I told you this is a very important compound in pyrimidine nucleotide biosynthesis. So, this orotate is synthesized with the use of enzyme dihydroorotate dehydrogenase, because here is the production here is the dehydrogenation process happening and dehydrogenation of dihydroorotate is happening. So, the enzyme is dihydroorotate dehydrogenase. As we can see, there is an incorporation of energy molecule, that is NAD positive, which is then converted into NADH plus H positive. Till now we have seen there is an incorporation ADP here in the very first step, then there is an incorporation of energy molecule here, that is NAD is converted into NADH into and H positive, and we have our orotate here. This orotate will be converted into orotidine monophosphate. Now, I told you that this orotate is very important, and after orotate we already have our pyrimidine nucleotide somewhat, because in the later on steps we will have our proper pyrimidine nucleotide. So, we have now our orotidine monophosphate because we have incorporated a phosphate group here. That is why we are saying orotidine monophosphate. If we were incorporating 2 phosphate groups, then it will be orotidine diphosphate, and likewise if we were incorporating 3 phosphate groups at once, which is difficult kind of, that is why we are preparing monophosphate, and then later on we can add on our phosphate groups. So, here we have prepared our orotidine monophosphate with the enzyme orotate phosphoribosyl transferase. We heard this name earlier also, PRPP. that is phosphoribosyl pyrophosphate. We have heard this name in the purine nucleotide biosynthesis also. It is very important enzyme, you can say. It is a very important element to synthesize our nucleotides. So, here there is an incorporation of PRPP and the enzyme orotate phosphoribosyl transferase. We have our orotidine monophosphate, in short we call it OMP. This OMP will then converted into uridine monophosphate. Uridine monophosphate is compound which is synthesized with the enzyme OMP decarboxylase. OMP is orotidine monophosphate decarboxylase, because it has came from this orotidine monophosphate. And in the previous step, that means in this step that is in when we are converting orotate into orotidine monophosphate, in this step specifically we have incorporated a ribosyl 5 phosphate, this. Okay. So, as I have told you that from the very starting, from the first step, we are not incorporating any ribosyl 5 phosphate, or we do not make our pyrimidine nucleus on ribosyl 5 phosphate. We will incorporate in the end, and we are incorporating it almost at the end here. So, this thing needs to be remembered, that after the step when we synthesized our orotate, in the next step, that means when we will convert this orotate into OMP, that is orotidine monophosphate. Monophosphate came, the name came because we have incorporated our ribosyl 5 phosphate here in this step only. So, this orotate is very important, and after that, because we are incorporating ribosyl 5 phosphate here, this step is also very important for us. After OMP synthesis, we have synthesized our uridine monophosphate by using OMP decarboxylase enzyme, and once we are having our UMP, that is uridine monophosphate, we can synthesize our di, tri, tetra, likewise our pyrimidine nucleotides. So, by using our uridine monophosphate, by incorporating one ATP molecule, that means we are giving one ATP to the step, and it will go into its in the next level, that means in the next standard, that is from uridine monophosphate, it will convert it into uridine diphosphate. Here it comes, because we are incorporating an ATP molecule here, we are giving the reaction one more ATP molecule. So, it will gone from uridine monophosphate to UDP, that is uridine diphosphate. Once we have uridine diphosphate, we can do several reactions here, like the first reaction is here, that is ribonucleotide reductase. By using the enzyme ribonucleotide reductase, you can convert UDP into deoxyuridine monophosphate or DUDP. When we are using this deoxy word, we do not write it capital D, we write it with small d, that is small d and capital UDP. So, it will be converted into deoxyuridine diphosphate with the use of enzyme ribonucleotide reductase. Once we have our DUDP, then we will convert it into, likewise we can convert it into DTMP by the use of another energy molecule and THF. THF is again here very important. There it was N10 formyl THF in the purine nucleotide. Here in the pyrimidine nucleotide, we have N5 N10 methylene THF. N5 N10 methylene THF, that is tetrahydrofolate, and finally we have our DTMP. Okay. Then DTMP is thymidine monophosphate, yani deoxythymidine monophosphate. Okay. and with the enzyme we made this DTMP with the enzyme thymidylate synthetase. Okay. Sorry, it was deoxythymidylate monosynthesis made from our thymine. Okay. And this UDP on the other hand, on the right hand side, this UTP by using an enzyme kinase will convert it into UTP and then at last CTP. UDP, from UDP we have synthesized UTP. How it will happen? Only by incorporating one more ATP here. That means we are giving the reaction one more ATP. So, it came from this level to the next level, from UMP to UDP to UTP. Okay. So, from monophosphate to diphosphate and then triphosphate. Here we are producing from UDP, uridine diphosphate, to UTP by incorporation of one more ATP. And then from UTP we can convert it into any other, especially cytidine monophosphate we are preparing here, another pyrimidine nucleotide. Okay. That is cytosine. From cytosine we have prepared our cytidine monophosphate, and what we are using enzyme here, that is CTP synthetase, cytidine triphosphate synthetase enzyme we are using, and we have our CTP, and here we are incorporating one of our amino acid, that is glutamine, which is converting into glutamate, and we have our final CTP. Let us revise this once more time with the steps here now. So, glutamine first of all transfers its amido nitrogen to carbon dioxide to produce carbamoyl phosphatase. That means, in the first step there is a production of carbamoyl phosphate. This reaction is entirely ATP dependent, and there is a enzyme which is working here, that is CPS2. See, there is an enzyme CPS2, carbamoyl phosphate synthetase 2, very important enzyme because this will help in the regulation of pyrimidine biosynthesis. This particular enzyme will help in the regulation of pyrimidine biosynthesis. CPS2 is activated by ATP and PRPP. We have studied about PRPP earlier, that is phosphoribosyl pyrophosphate, and it is inhibited by UTP. That means, once we have our UTP completely, that means we have completed our reaction, once we got our UTP, then there will be the limit of this particular enzyme CPS2, and there will be a feedback inhibition happening here. Second step, that is carbamoyl phosphate synthetase 1, that is a mitochondrial enzyme and which synthesizes our carbamoyl phosphate from ammonia and carbon dioxide, and in turn which gives urea. Prokaryotes have only one carbamoyl phosphate synthetase, which is responsible for the biosynthesis of arginine and pyrimidines. Arginine again is an amino acid. Carbamoyl phosphate condenses with aspartate and forms carbamoyl aspartate, because we have incorporated aspartate here. This reaction is catalyzed by aspartate transcarbamylase, and dihydroorotase catalyzes the pyrimidine ring closure, and there will be a loss of one water molecule, which then produces, there are three enzymes which we are working here, that is CPS2, aspartate transcarbamylase and dihydroorotase. These are the domains, that means the functional unit of a single protein. This type of protein will be called as multifunctional enzyme. That means a single protein that is a domain, a functional unit for three different enzymes, that is CPS2, aspartate transcarbamylase and dihydroorotase. This type of protein, this type of enzyme will be called as multifunctional enzyme. Okay. So, in the next step, the pyrimidine synthesis is an NAD positive, that is NAD is again energy molecule. energy molecule we are giving here for the formation of orotate. I told you this orotate is very important. Later on we will discuss what will happen if there is a misfunctioning of this orotate step. There will be diseases because of it, misfunctioning and deficiency, accumulation, what will be happen, we will discuss later on. So, the next step in pyrimidine synthesis is an NAD positive dependent dehydrogenase leading to the formation of orotate. Then after this step, very important thing is happening, there will be an incorporation of ribose 5-phosphate. Once you get your orotate, now we need to incorporate our ribose 5-phosphate, and the thing we will get is our orotidine monophosphate. Name suddenly changes from orotate to orotidine monophosphate. Now there is a word called as monophosphate here, and from this step we will synthesizing our pyrimidine nucleotides. That is, the reaction is carried or catalyzed by orotate phosphoribosyl transferase. It is an enzyme comparable with HGPRT. HGPRT is hypoxanthine guanine phosphoribosyl transferase. If you do remember, we studied this particular enzyme in the previous lecture in the purine nucleotide biosynthesis lecture, and there was a disease related to this particular enzyme, that was Lesch-Nyhan syndrome. That means, if there is a deficiency of HGPRT, there will be a disease called as Lesch-Nyhan syndrome, which we studied in our previous purine nucleotide lecture. For more details, you can go through the lecture as well. Now, in the next step, OMP will undergo decarboxylation and form uridine monophosphate, that is UMP. Orotate phosphoribosyl transferase and OMP decarboxylase, they are the domain again from a single protein. There will be a defect in the bifunctional enzyme causes orotic aciduria. This I was talking about. If there is a malfunctioning in the step with the enzyme, then there will be a disease called as orotic aciduria. And by an ATP dependent kinase reaction, UMP is converted into UDP, which serve as a precursor for the synthesis of DUMP, DTMP, UTP and CTP. Once you have your UDP, you can go for the synthesis of DUMP, DTMP, UTP and CTP. Then ribonucleotide reductase converts our UDP into DUDP by a thioredoxin dependent reaction. Then our thymidylate synthetase enzyme that catalyzes the transfer of methyl group from, I told you that we have a different THF here, that is N5N10 methylene THF, and it will produce our DTMP, that is deoxythymidine monophosphate. And UDP undergoes an ATP dependent cycle to produce UTP. Cytidine will synthesized from UTP, that is the last reaction, and CTP synthetase is an enzyme and glutamyl provides the nitrogen. So, this was all about our steps. Now, the regulation which I was talking about, regulation, there were two enzymes actually, we are going to talk about three enzymes here. In bacteria, aspartate transcarbamylase, that is ATKase enzyme, is this step is a committed step. This is very important step which control the enzyme as feedback mechanism for pyrimidine biosynthesis on the end product, that is the production of CTP. In certain bacteria, UTP also inhibits ATKase, but it is not everywhere. But we can see that there is incorporation, there is this step in some bacteria that UTP inhibits our ATKase. ATP however stimulates ATKase activity, and the two enzymes which I have told you when we were studying about the synthesis, that is CPS2 and OMP decarboxylase. These two enzymes regulate our pyrimidine biosynthesis. Okay. So, CPS2 is the regulatory enzyme pyrimidine synthesis in animals. It activates PRPP and ATP by inhibiting the UDP and UTP, whereas OMP inhibit UMP and CMP. Finally, you can see through the regulation, you can see the line diagram which shows the regulation, that is glutamine goes into carbamoyl phosphate synthetase, then which converted into carbamoyl phosphate, then are orotate, then OMP, then UMP, and then there is a production of UTP and CTP, and this UTP ultimately stops, which rather I would say feedback inhibit the carbamoyl phosphate synthetase enzyme, and this whole process will be regulated with that. This is the degradation. The last degradation is the pyrimidine nucleotide undergoes the similar reactions like your purine nucleotide. What reactions are here? Dephosphorylation, deamination and cleavage of the glycosidic ring. Once these reactions will happen, there will be a degradation of your pyrimidine nucleotide. The last products which are produced, that is beta alanine and beta amino isobutyrate. These two are the products which are produced as the degraded product of pyrimidine biosynthesis, which is which is finally produce acetyl coenzyme A and succinyl coenzyme N. You can see through the structure, you can see through the line diagram here that last product, that is your beta alanine and beta amino isobutyrate is produced, that are the degraded product, and lately it will be converted into acetyl coenzyme A and succinyl coenzyme A. Thank you so much. These were the references from the topic. Hope you enjoyed learning. Thank you so much.